Hi everyone. We're going to finish up our discussion on translation and then talk a little bit about mutations. Okay, just to wrap up, review what we had talked about earlier. When we finish transcription in the nucleus, we have the DNA that's transcribed into mRNA. And remember, there's three modifications that occur. We add on the 5' cap, we add on the poly A tail, and both of these are to protect the mRNA and help the mRNA to be recognized by the ribosome. And then we also remove these introns. And the introns are just sequences of DNA that don't actually contribute to the protein that you want to express. So at the very end, we have our mRNA, and this is what leaves the nucleus and enters into the cytosol where it's going to bind to a ribosome. And so the next part of the process is how this mRNA becomes a protein. Translation can also be divided into initiation, elongation, and termination. And those steps should sound very similar to the same steps that occurred during transcription. So we're going to go a little bit more in depth with this. So the initiation part of translation starts at the very beginning of the direct protein sequence. So notice that the beginning of the mRNA nucleotide sequence doesn't actually start with the proteins. You have the 5' cap, and then you have just this bluish part right here, which signifies the part where it just helps the mRNA to bind to the ribosome, but it doesn't actually code for any important proteins. So the ribosome is going to bind, and then it will start initiation right here at the very beginning of the green. So during initiation, the first thing that happens is you have the mRNA, so the sequence, binds to the bottom half of the ribosome, which is the small ribosomal unit. So it looks like kind of like a half of a hamburger. So it binds there, and then the tRNA base pairs with the start codon. So this is the very beginning of the protein chain, and so we have the mRNA sitting on top of the ribosome, and then the tRNA comes and it matches its anticodon with the start codon, and that's the very first part of this process. And then after this binding process happens where the tRNA matches with the very beginning of the mRNA on the bottom part of the ribosome, then the top part of the ribosome comes and attaches itself. So that's when it's called the large ribosomal subunit, and it will attach on the top. And then this creates a functioning ribosome. So after initiation, there's elongation. Elongation can be broken up into a series of steps. So after we have this, the tRNA binds, there's 3 different sites on the ribosome, and your textbook only talks about 2 of them, so here I'm going to talk about all 3 sites. In the middle is called the P site, and this is the site where your tRNA is sitting that has attached to it the growing polypeptide chain. So here it says that the tRNA attaches to the growing polypeptide chain, which is in the P site, which is what we said. And so what's going to happen next is that the new tRNA with the next amino acid on it is going to come in and bind on the A site. So it's going to sit right next to the P tRNA with a growing chain, and it's going to sit right next to it on the A site. And when it sits on the A site, its anticodon in the back on the bottom of the tRNA is going to bind to the codon on the mRNA chain. So after this happens, we have the P tRNA and then the A tRNA. They're sitting right next to each other, and there's going to be a transfer process where the long polypeptide chain that was on the P tRNA is going to get transferred onto that new amino acid that is on the A tRNA. So the whole chain gets pretty much shifted over. This requires some energy, and you can see in this little picture that there's a couple of GTPs that are involved in the process. Okay, afterwards, the P site with the tRNA that now lacks an amino acid is going to shift over to the E. So this one right here, it shifted its whole polypeptide chain onto the A tRNA, right? So now this P tRNA is empty, and then it's going to shift over to the E site, and then it gets ejected off of the ribosome. Also, the tRNA that was on the A site, which now has the longer polypeptide chain, gets shifted over to the P site. And then after this, the process continues to go on and on. Now the previous A tRNA is now the new P tRNA, and there's going to be a new tRNA that comes over here, and then the polypeptide chain will shift, and then after it shifts over to the A tRNA, both the P and the T, both the P tRNA and the A tRNA will shift over to the E and P sites, and the empty one will now leave again. So this process goes over and over again until termination happens, which is when the elongation stops when the ribosome reaches a stop codon. So there's very specific codons, UAA, UAG, and UGA. You don't have to memorize that, but just realize there are stop codons. And so when the stop codon is found, then the ribosome stops translating. So just to review everything we talked about, transcription is when DNA goes to RNA. And remember that the mRNA is processed by a 5' cap poly A tail, and then we remove or excise the introns. And then in translation of the RNA to make proteins, we have the initiation, which is when the small bottom part of the ribosome gets attached to the mRNA, and then the tRNA goes onto the start codon, right? And then afterwards, the top part happens. Then elongation, just know that there's E, P, and A sites. The long chain starts in the P site, and then will shift over to the A site, and then the two tRNAs will shift over to E and P, with the empty tRNA leaving, and until it reaches the stop codon for termination. So just some notes that we haven't mentioned yet about translation. This process is very fast, and so you can, even though it sounds kind of complicated, this whole thing happens to make a whole polypeptide in like less than a minute. Also note that one mRNA can be translated by multiple ribosomes at the same time. So you could think of it as like a long ribbon of information and like a bunch of little ribosome hamburgers on top of on the same chain and as the mRNA just keeps on like shifting through. This allows the cell to make multiple proteins of the same type very fast. As soon as the polypeptide chain starts to grow with the amino acids that are constantly being linked to it, it instantaneously starts to fold. And so the three-dimensional structure or tertiary structure of the protein begins as soon as it's being created. Okay, now to talk about mutations. We've talked about the whole process assuming that things go well. But mutations are when there's a change to the nucleotide sequence of a cell. This can either happen in two major ways. You could either have a substitution, which you can see in this picture is just when one amino acid switches to a different kind of amino acid. Or you can have insertions or deletions. And that's when, just like it sounds, an insertion is when a nucleotide that shouldn't be there is placed in between, in the middle of the chain. And deletion is when one of the nucleotides that should be there is removed. So to get more in depth with substitution mutations, you can have something called missense mutations. And this is just when an amino acid is changed into another amino acid. So if you look at this picture right here, here's a simple example where you should normally have an AT pair, but instead it gets switched to a TA, and that changes what ultimately is read for the mRNA, and then that changes a GAG into a GUG, and that codes for a different amino acid. And so you can see that there's a change in one amino acid, and although it sounds small, a change in one amino acid can actually totally affect the structure of a protein. In this case, you could, for sickle cell anemia, the shape of your red blood cells is very much affected by the change of just one amino acid, and actually changes it into this like weird crescent shape. It's a very painful disease to have. So aside from missense mutations, another example of a substitution mutation is when one of the nucleotides is switched, but it doesn't actually change the amino acid that is coded. And so here's an example. It's normally supposed to be an A for the nucleotide sequence, but instead it's a T. And notice you can still get the same amino acid of valine. And this is because multiple codons can actually code for the same amino acid. So if you're lucky, the mutation may not actually affect the protein that's created. The last kind of substitution mutation you can have is called a nonsense mutation. And this is when the amino acid is actually changed into a stop codon. And so if you have this supposed to be, this is supposed to be like a C or something, cytosine, it gets changed to a thymine, and then this becomes a stop codon. and then that cuts short the polypeptide or the protein. Um, and so out of all three of these, obviously silent mutations are the least negatively impactful on your body, but nonsense mutations are probably the worst out of the three for substitution mutations because it has the most dramatic effect on the protein. Okay, the other kind of mutations you can have are called insertion or deletions. They're both different types of frame shift mutations. Frame shift mutations, which are due to either an insertion or deletion, causes the regrouping of all subsequent codons. And so when you insert a nucleotide into the sequence, because codons are read as groups of three, you're reordering all of the codons that occur after it. And that means you change all of the protein, amino acids in the protein. So for example, here it's supposed to be TC, and then if you insert an A in between them, now you're shifting all these down one, and so now the A gets regrouped with the CA, and the T gets kicked off into the new codon and now becomes TCA, and you see how each one of these becomes a new amino acid. So frame shift mutations usually are very harmful for your body and are much worse than substitution mutations. Okay, what causes mutations or mutagenesis? Mutagenesis is just the production of mutations. Sometimes these just occur randomly in your body. Sometimes your DNA polymerase II, which is supposed to be a proofreader, isn't so great and just doesn't do it quite right. Or the other thing is mutagens. So mutagens are actually external to your body. They're either physical or chemical agents that can cause mutations to occur. So for example, physical mutagens, X-rays and radiation. That's why if you sit out in the sun too long, you increase your risk of skin cancer because that UV light and the radiation can mutate your DNA. Also, chemical agents examples would be like benzene. Benzene is a really common solvent in chemistry. So if you go off to become like a major in chemistry, you might be using benzenes, and you should just be careful because that can actually mutate your DNA. So realize that almost all mutations are generally harmful for your body. Usually the DNA that you have codes for very specific proteins for a reason, but there's occasionally chances where your mutations may be positive. And this, of course, leads to natural selection and thus evolution of the species. All right, thank you so much for watching. Hopefully this all makes sense. Thanks, bye.